Thank you for joining us today for this webcast entitled Debating Interchangeability, Efficacy and Safety of Biosimilars in the Management of Inflammatory Diseases. I'm Dr. Len Calabrese. I'm a professor of medicine and vice chair of the Department of Rheumatic and Immunologic Diseases here at the Cleveland Clinic. And today we're going to review some of the highlights from two CME activities on biosimilars developed by CME Outfitters. You can find these and other CME activities on biosimilars on CME Outfitters Biosimilar Hub. By the way, please follow me on Twitter. I have a lot to say about this field and the broader range of rheumatology. So let's begin this webcast. So before we get into the questions, let's just briefly review these objectives we went through. First learning objective was distinguishing biosimilars from generic agents in the management of inflammatory diseases or immune-mediated inflammatory diseases. So to do that, you have to be able to answer the question, what is a biosimilar? A biologic that is highly similar, and I emphasize this highly similar, it's a word that's like a mantra in the world of biosimilar drug development, to the reference product with no clinically meaningful difference in terms of safety, profile, purity, and potency. Biosimilars are not generics. And this little table kind of tells us the difference between biosimilars and generics. I think that's kind of intuitively obvious. Generics are copies of small molecule drugs. They're carbon copies, if you will. These molecules are simple and well-defined. They're predictable chemistry, easy to characterize, and relatively stable. They have a low potential for immunogenicity. On the other hand, biosimilars are monoclonal antibodies, at least the ones that we will be talking about today, can be other biologically or recombinantly produced molecules that are large, often 150,000 kD or more, complex structures with secondary, tertiary, and quaternary relationships. They are manufactured via recombinant technology and unique cell lines. A lot of this is proprietary. They're difficult to totally characterize. They're sensitive to storage and handling conditions, and because they're complex proteins, they have potential for immunogenicity. Learning objective 2 is to summarize interchangeability of biosimilars with the reference biologic and how it impacts on clinical practice. So there's a term, interchangeability, which is seen throughout the biosimilar debate. And, it's a word that we use in the lexicon all the time. Something's interchangeable, we just, switch it with something else. That is not what interchangeable means in the biosimilar world. It is an official FDA designation that has rules and regulations which allow it to be entered into the marketplace. If a drug is interchangeable, a biologic is interchangeable, it may be substituted for the reference product without the intervention of the healthcare provider, that's you and me in terms of physicians, who prescribe the reference product. To achieve this designation of interchangeability, it has to go through a rigorous, high-impact process, different from just biosimilar approval. Obviously, it must be biosimilar and have this highly similar pattern of fingerprint designation, and it must have an acceptable safety profile. At the moment, the clinician can make a change in a medication, regardless of whether it's interchangeable designation has it or not. And currently, a pharmacist cannot make a switch between a biologic without a prescriber approval, because at this moment, and this is important, I emphasize this again, there are no approved interchangeable products. So we're talking about a process and what may be coming down the pike. Thirdly, we debate and evaluate the safety and efficacy of emerging biosimilars in the treatment of inflammatory diseases. And there's a lot of data going on, particularly in the treatment of immune-mediated inflammatory diseases, or IMIDs, showing efficacy and safety, PK, immunogenicity, and the like. If we look at just clinical trials in these IMIDs, for the most part, we have seen that most of the trials done to date have been performed in rheumatoid arthritis, psoriasis, and psoriatic arthritis. Based upon this, extrapolation goes to other indications, including inflammatory bowel disease. And the extrapolation principle supports the use in all the indications for which the originator was approved. So etanercept is approved in psoriasis and psoriatic arthritis and SPA, but if the original study is in rheumatoid arthritis, it received all of the approvals. Similarly, infliximab plus its approval in inflammatory bowel disease. This has created some consternation among some providers, but thus far it has been the standard. A question on all of our minds in terms of providers and patients is this notion of switching. If I'm on an originator drug and for some reason I'm being incentivized by my insurance company to save money, if I take a biosimilar, what are the data that shows that it will remain effective and non-toxic to me? And here we're seeing the NOR-switch study. This is a large study that encompassed multiple IMiDs, and basically people were placed on the originator infliximab, and over time all were switched to biosimilar infliximab. And then the primary analysis was looking for efficacy. And as you can see from this bar gram showing the confidence intervals, to the left would favor infliximab, to the right would favor the biosimilar, and all of these lines cross zero, some barely and some robustly, but it was designated that the biosimilar, after this large nationwide switching program, had similar efficacy across indications. They also looked at safety and some biomarkers, none of which dissuaded the conclusion that this single switch study demonstrated enduring efficacy of that switch with acceptable safety. CT-P13 is a biosimilar, and you'll see the DYBB, that is a designated label for this in particular biosimilar. So since there are now multiple biosimilars of infliximab, we have to have a way of sorting this out. It was approved by the FDA in the spring of 2016, and here are some of the summary data that have been shown in rheumatoid arthritis, spondyloarthritis, and the switch study that we were referring to. All of them showed a highly similar efficacy with core disease indices, in particular ACR scores, DAS scores, biomarkers for rheumatoid arthritis, for ankylosing spondylitis, ASAS scores, which are used in virtually all trials, plus additional markers of quality of life and motion, such as BAS-Phi and BAS-Me, as well as chest expansion. And in the Finland study, actually additional measures such as quality of life measures, such as fatigue scales. Again, this is a recurrent theme in this literature. Drugs that have moved into the biosimilar acceptance range all have met this standard of being able to demonstrate this highly similar efficacy. I will kind of close in and mention this EGALITY study. This is a study that just was published a few months ago on the British Journal of Dermatology. This is a unique study. This was starting with the originator, etanercept, then switching to biosimilar etanercept, GP2015, and the index was looking at the effect on the PASI score, the skin score. And as you can see, per protocol data show that there's really no difference between the biosimilar and the originator. What is not shown on this slide was that this study then went on to make two subsequent switches from the originator to the biosimilar, and then the biosimilar back to the originator with no apparent loss of efficacy or safety. So this is the type of study that we will see more of moving to the future. Toxicity data has to be similar between the biosimilar and the reference product. This is the thing that patients care most about, doctors care most about, and we will continue to have a sharp lens focused on this. So to conclude this, biosimilars are not carbon copies. So there are reasons to have a questioning mind, but thus far, the data that I've shown you, which are representative of the larger aggregate database, suggest this, quote, highly similar, end quote, picture of efficacy, safety, supported by PK/PD, immunogenicity, and a plethora of ex vivo data. I already see a ton of questions coming up, and now we can kind of dig in and go to town on this. Without further ado, I am going to dig in to the questions. We have a bunch of questions here. All right, first question, I think relates to the Northwich study. And if you're really paying your attention and doing your homework, you saw that those confidence intervals differed for different diseases. Remember that this study was not powered to look at the differences between individual disease. It was an aggregate power for equivalency, and it met it. Furthermore, if you compare diseases, to be able to critically assess whether psoriasis improves, this is done with great precision and accuracy because we can measure this. Diseases that rely on a lot of subjective measures like IBD often will have much broader confidence intervals, and so that's the explanation for that. All right, so we have a question here. Can the pharmacist change my order to a biosimilar without my permission? I live in Ohio. Well, at the present time, the only way that would happen would be if there were an interchangeable drug, and as I told you, there are no interchangeable drugs right now, so you are the decider here of whether the patient gets this. And I'll come back to a little bit of embellish that in a bit. Next question, is it true that biosimilars may only have approval in one indication, but then can be used across various diseases without further testing? All right, so I get exactly what you're saying. I tried to engage on this. It is not that they only have one indication, but what they are required to do is to do a pivotal trial in at least a single disease that the originator is approved for, and if it meets the burden of proof of efficacy, then by the term we call extrapolation, it receives indications in the other drugs. And if you actually look through this, I think there's a knee-jerk response to say, well, geez, psoriasis isn't rheumatoid, or inflammatory bowel disease isn't spondyloarthropathy. Wherever the disease has been selected thus far for these approved biologics, they have demonstrated equal or highly similar efficacy. Now, I sit on a P&T committee. I'm not convinced the savings are significant enough to switch stable patients. Is there any real cost savings? All right, this is a fabulous question. And, if I were sitting with you, I'd probably say we have to go to the bar so I could drink some white wine and wave my hand to answer this, because there's a lot to this. Let's break this down a little bit. We know that in countries in Europe that are single-payer systems, particularly countries such as Finland, who have doubled down on it, that the saving to the healthcare system by mandating the use of a biosimilar has been over 50%. So, you're looking at this, and you know how much these drugs cost, and this is an entire country. This is big money. Well, this isn't Finland, and the way that we fund healthcare is, in my opinion, bug nutty. It's a patchwork quilt of, what is available, what are my resources, how many millions of people do we have that aren't covered. It's not an optimal system. I think all of us would agree, no matter where you fall in the healthcare debate. These drugs will have a cost savings, and they will be sold for below the marketable price of the originator. And the originator now can double down, and they can renegotiate their own price. And, we have these managers that negotiate price. So the question that you're asking, who is going to save the money? Who is going to save the money? Well, to me, my personal decision is based on two components. I have a patient, they are doing good on an originator. What would be the incentive to have them switch to a biosimilar? They will do no better than being on the originator. And while the safety signal looks robust, I am subjecting them to some degree of risk by switching them from what they were doing great on? Well, I will do it under two circumstances. Number one, that they demonstrate to me that their provider has incentivized them to switch on a financial basis. I haven't seen this happen yet. I'd like to come in and say, I don't have to make a copay anymore if I switch to this biosimilar, or I'm going to get a rebate, or something is good going to happen to me financially. Then we can work through this on a shared, informed decision-making process and make a decision. Or the second matrix would be that my healthcare system, Cleveland Clinic Enterprise, has negotiated a deal with biosimilar or biosimilars and comes to us as a department and says, if we can transfer some of our biologics to the use of biosimilar X, Y, or Z, our healthcare system will save, X millions of dollars and improve our bottom line and our stability. And, this has transparency that we will be able to meet our mission as an institution in a better way. If I can't demonstrate that my healthcare system, or you can insert your practice, or your patient will clearly demonstrate improvement, and that the savings is some nebulous savings to a large provider or a benefits manager, then I'm not in favor of switching either. As we say in the world of evidence-based medicine, that is C-level evidence, my personal opinion on it, and that's where we would go. So there's a series of questions here that kind of follow along with that. Would I switch one of my stable RA patients? Yeah, give me two of those variables. Going to save them money or save us money, and I will engage with them to discuss this. It's a much easier decision on a new patient. Why, if you give me drug X costs 50 units and drug Y costs 100 units, and I'm just starting out, and that's an easier decision to make. It's a cost savings around. But switching patients, there has to be an incentive coming down the line. What are my thoughts about physician perceptions of biosimilars and how interchangeability will affect this? Would it be fair to say that physicians already see biosimilars as similars to the originator? Will interchangeably increase the adoption of biosimilars in your opinion? Do you think that these views are specific to the inflammatory space, or is it going on in oncology as well? That's a great question. Since we don't have an interchangeable drug yet, there are studies that have been filed to do this, but we don't have the interchangeable. They remain, at the top of the mountain. The bottom of the mountain, the free kicks at the ball here are starting a patient on a biosimilar versus an originator. The second hardest is making a switch from a stable originator to a biosimilar. The third and the peak of the mountain is the interchangeable thing. I think we'll cross that bridge when we get there. I would think that pharmacists that I've talked to would want to maintain interprofessional communications with providers despite the letter of the law. And I think that the rest of this question, that what physicians already see biosimilars as, I think from my discussions and my work and my website, my Twitter feed, some of the data I've seen that Jack Cush has shouted out, I think most of us are pretty comfortable of the concept. I think the big questions are, where's the money going to go, and what will happen with interchangeability? All right, another question. What about drug-drug interactions? Highly similar concerns to me when it comes to drug-drug interactions. Biologics, in terms of DDI, most of this is really in terms of altering the inflammatory state and subsequent downstream metabolism. I don't really have much in the way of strong concerns that the DDI of a biosimilar will be dissimilar to the originator. My biggest fear coming into the biosimilar world has been the issue of immunogenicity. My field, I come into this as a basic and translational immunologist, and I know that I cannot make two antibodies that are identical to each other because of the massive post-translational modification and how this affects a lot of the immunologic behavior of a molecule. At the same time, we know that all of these drugs are immunogenic, and now that when we give them to normal volunteers that don't have inflammatory diseases, virtually everyone will make antibodies to it. On the other hand, the data have been pretty reassuring that the immunogenicity profiles are, again, quote, similar, highly similar, between biosimilar and originator. I think that everyone probably appreciates if you're on the originator and you have immunogenicity to it, well, then you can't use the biosimilar because you'll have immunogenicity to that. And I think everybody's got that in their head right now. So let me see if I can squeeze in one more question here. Are these biosimilars going to be mandated in the U.S. under the auspice of being value-based care or quality? I have a major concern what these agents will mean when it comes to efficacy in the real world of patients with various other comorbidities. You know, I think that's a good question. It's meritorious. I have no idea what's going to be mandated. You know, we all wear different hats. You know, if you're a private practice and you stock drugs for infusibles, maybe you have much more command over this. The great swatch of clinicians who take care of IMIDs, who work for group practices or large mega group practices, you know, we're not the deciders all the time. And P&T and those that negotiate costs are going to be engaging with us. I will tell you, as of this date, you know, October 10th, 2017, we have not had a major granular conversation for this large 30-plus group of rheumatologists here. I know that it is being weighed, and I know that it's going to come. So, you know, we're still at the beginning. I think we have a lot of data poised to help us. But I think the next time we get together for these conversations, we're going to be able to tackle these in the real world. So, you know, I'm sorry I didn't get to all these questions. Again, reach out to me on Twitter, capital L, capital C for Calabrese, capital D, capital O. Reach out through CME Outfitters, through email. I'm pretty easy to find at the Cleveland Clinic. So thanks, everybody. This has been a fabulous opportunity to engage with some really insightful questions. Come back to CME Outfitters, again, for a lot of other great programs, and we look to interact with you in the future. Thank you.